Hello everyone, welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about string database. So this is the link for this database and this is the interface. And this database is widely used for biological purpose like protein-protein interaction. And this is the web source that provides comprehensive information on protein-protein interaction, including both physical and functional associations. So here is the search bar. Here, for example, uh, I want to see the interaction between the PDL1 and some other protein partners. So this is the drop down. Here you can select different organisms, and I want to see the in the human. So there are different protein interaction. So here you can select your target organisms, okay, and then select. So I want to select here the Homo sapiens, and then select and then search. So here you can see that this is the CD274. So when you click here, you can see this is the PDL1, programmed cell death 1 ligand 1. So this is the PDL1. So this is with this target protein, it will show some other interaction with some other protein partners, okay. And this kind of interaction are direct, like physical interaction, and also indirect interaction. And this database, it will show the both the evidence based or the experimental based, and as well as computational based. So these both kind of like prediction and also experimental interaction also it will show. So here the different protein partners, you can see it interaction, and here is the settings, and here you can, there are different parameters, so you can select accordingly what you need. So as you see here, this that line, so this line actually, this the thickness of this line, it indicates that how strong this interaction, and these are the evidence based or experimental based, okay. So like this you can see, we know that PD1 and PDL1 interaction, this is very strong, and this already experimentally proved, right? So you can see here the PD, you can see first the PD1 protein. So this is the PD1 protein, I guess. So if you select here, so you can just click it to check it out. So here is the PD1, program cell death protein 1. So this is the PD1 protein. So you can see this PD1 and PDL1 interaction, this is very strong interaction. This is why the line, the thickness of this line, this is thicker than the other interacting proteins. And when you click here, then it's showing the interaction between these two proteins. So when you read out this, then you can see that what kind of pathways are interacting and what are the different mechanisms. So it all in detail this showing here. And also in the below there are some different information, some important information, like here you can see the experimental or biochemical data. So experimental means that it experimentally proved, or here are some publication that that's showing that there are some interaction, physical interaction. So here this is why yes. All right. So now in the settings you can select different parameters, like this is a confidence. So this confidence score, it indicating the stronger interaction or the weaker interaction. So here is a medium confidence. So when you switch to the high confidence, then it will show some kind of strong interaction. And update, so when you select the update, then there are some proteins will be eliminated. So here it will show only some interacting proteins, those are strongly interact. And also if you, I prefer to select the highest confidence. So when you select the highest confidence level, and also if you select no more than 5 interactions also, that also fine. So if you select this no more than 5 interaction, that means that the strong interacting protein partners only it will show, and other protein partners all are eliminated. And then update. So now we can see these 5 protein partners, these are very strong interacting proteins. So my target proteins is the PDL1. Okay, so now we can see these PDL1 proteins. So it interact with the CD28, it interact with the CTLA4 PD1, and it has the weaker interaction with these proteins. But when you click here, you can see what kind of interaction it is. So here also it is showing that experimental none. So that indicating that there is no experimentally based any proof or any kind of publication to show to prove this interaction. So there is no evidence. So this interaction is, so this protein and this protein interaction, this is completely prediction, and this is the computational based. There is no experimentally based any kind of evidence. And also you can, if you select this protein and this protein, you can select also this. So here, so if you select here, then it is showing that the experimentally proved. So yes, so this protein and this protein interaction also it experimentally proved. Okay, so there are different kind of information. So there are different kind of the application for this protein interaction, and you can use it in different purpose, like drug discovery, target identification, and also different protein uh, interaction. And some kind of protein interaction are if you try to select or if you try to predict, there are some model, some protein interaction model. So if you want to predict beforehand, then also you can use this tool to predict this kind of protein interaction, and then you can select the model based on that prediction. So this kind of, in this purpose also you can use widely. And I also saw something like as I showed here, that this is the information that's showing the this is the interacting proteins. But when you show here, then it can show something that is some kind of data, some kind of database, like it's showing uh, some information regarding it. So here it's showing that protein interaction intact and detected by surface plus bond and resonance assay, average detection confidence high. So let's click this one, and then you can see based on that there are some kind of publications. So that publication, it proved this kind of interaction. So this is also, you can see, if you want to see in detail like what kind of pathways or how it is interacting, then you can go through this publication. And back also there are different, so you can see, for this is why it is very strong and this line are thicker, because there are a lot of paper and a lot of publications that prove that it has some kind of physical interactions. So also here you can see the PDB entry structure complex, and here also this is the structural based and crystal structure. So if you also click here, so you can see this is also this is the PubMed ID, and also this is a paper. Okay, all right. So and here also if you want to export this interaction, then also you can go through. So you can go to the back. So here you can see this interaction. So if you want to export this interaction, then also you can do here, like download the PNG format and different formats. So you can download from here, so you can use this interaction in your publication. Okay, so this is very useful. So and also I want to show that So if you click also in the more option, then you can find there are also some kind of computational based interaction. So you can see there are a lot of protein partners here. So these protein partners like this kind of interaction, so you can see this is very thin line. So this thin line indicating that there is no experimental based any kind of proof. So you can, let's see here, so you can select, you can see, okay, so here is the experimental, yes. So that means that only one paper is here, I guess. So let's check it out. So here you can see, you can, if you show a button and then see that how many papers are there. Yes, right. So that only one line that indicating that only one paper is there. Okay, so if you click here, no, it's not, it's there is no papers attached here. I think you need to check in the protein data bank, or you can go through this paper. Okay, so back, you can check the another one. Maybe that these all are this thick line, this thin line. There is not that many interaction based on the experimental any kind of probe. So also here in the analysis section, so if you select this analysis, then you can see there are different pathway you can select, like this interaction. So let's say I want to go back, and only I want to select some 5 or 10 protein interactions. Okay, so confidence level, I need to go, here is the options, back, okay, here settings, okay, so here is the highest confidence level. So here I want to show that if you want to go to the analysis part, and then you can see there are different pathways, like which kind of pathways are associated with these interactions. So here you see the biological process. There is the T cell activation, T cell co-stimulation, and also these are immunology part. And here is the cellular component, and what kind of cell surface, external side of the plasma membrane. So these are also associated with this interaction. And also it's showing the strength, so strength indicating that if it is the strength is more, then this is the stronger associated with this kind of uh, protein interaction part. So that means that like 2.33, so this is the T cell co-stimulation, so this is very strong. So that indicating that this interaction, it is highly regulated in this T cell co-stimulation part. But if you compare with the other part, like negative regulation of the T cell activation, or the regulation of the T cell activation, in this kind of case, their role is lower compared to this T cell co-stimulation part. And also there are some other references, like a disease gene association. So in the disease gene association also you can see the autoimmune disease. So this is strength is 1.73. So in the autoimmune disease also this interaction, it plays an important role. So in an important role in it play, and also in the renal cell carcinoma. So in the renal cell carcinoma, this interaction also, it, like if you click here, then you can see that what is showing here. To click here, then you can see something. Okay, let's load this page. But if you go further down, then you can see this kind of like how many associated pathways are there. So there are different pathways like negative regulation of the T cell activation, negative regulation of the immune system, and and also this part it's showing the how strong this is. So this is very stronger interaction. So negative regulation of the T cell activation, we know that PD1 PDL1 interaction, this is it influence the T cell functions. So this is why negative regulation of the T cell activation, this is very higher. So this is the highest in this part. This interaction it highly regulated in this part. But where in the regulation of the T cell proliferation, this interaction is not that much associated. So this is why it is very lower. So it is only 2.6, and here is the 3.8 around, right? So let's see here the disease. So this is the disease part. So disease, it contains like human genes of the renal cell carcinoma. So the renal cell carcinoma, here there are some kind of protein partners. So you can see this BTL1, that is our target proteins. So associated with these protein partners like CTLA4 and some other protein partners, these all are associated in this renal cell carcinoma. This is what it is, all are listed here. Okay, so so this is the part. So there are a lot of things here, and also there are some kind of database that are similar to this kind of database, that is like Reactome and also BioGrid. So later I will show this kind of some kind of tutorial video on that kind of database. So here is the almost the functional part, that all I covered, that is most commonly used normally. So I hope this video will be helpful. So please watch this video till end, and if you like this video, kindly hit the like button and share it, and please subscribe my channel, and if you have any queries, kindly write in the comment section. Thanks for watching.